Thank you very much for having me here today. Good morning, everybody. So the title of my research that I'm going to present today is RNA sequence based transcriptional map of necrotic enteritis Clostridium perfringens type A strain. And I work in collaboration with John Prescott. So I don't have to talk much about the background of the disease. King One already told us. Let's go direct to what means the RNA sequence transcriptomics. RNA sequence transcriptomics is a quite new tool, is a powerful tool for understanding the whole transcriptomic map and also the RNA-based regulation in any organism. So it's a kind of bird's eye view of all genes in a specific condition at the same time. And also when we are talking about RNA sequence, we are talking about messenger RNA that plays an important role in making proteins. We are also thinking about no-coding RNAs and small RNAs that do not code proteins, but more and more we are seeing that they are related with regulation of the genes. So our primary goal is to understand and control necrotic enteritis in broiler chickens. So for that, we are going to construct transcriptomic maps for Clostridium perfringens from in vitro bacteria, that is the bacteria that is grown in the lab, like very controlled conditions, and also in vivo, that is from chicken intestinal loops that I'm going to show a little bit further. And the idea of the RNA sequence is to isolate these Clostridium perfringens from in vitro or from in vivo, extract and purify the messenger RNA, convert that to the cDNA, and then sequence, in our case, by the Illumina platform. So here we have what would be the chicken intestinal loops. We have the chickens that are kept alive during the experiment. They are kept anesthetized, and then after that, the surgeon is going to take the duodenal, in our case, and they're going to make 5 centimeters loops, and the cells, the perfringens or the saline, are going to be injected in each one of these loops. And then after 4 hours of experiment, the animal is sacrificed and the intestines I take. And then here in our left, we can see here, well, this pointer, right? This distended part. Yeah. Well, we can see that there is this distended part that is where the inoculated with the virulent C. perfringens that cause this gas and accumulation. And the other loops are practically like the same way. So here in the RNA sequence, this is that we are going to see with the output of the RNA sequence results. So in the top, these small bars are the small reads, sequence reads that we use to align to the genome DNA or to genome of the Clostridium perfringens, that are these yellow bars. So in this specific example, I just want to show the discovery of a new gene. So in between these 2 yellow bars, we can see here there is a, let's see if I can, oops, oh. So, let's see here. Okay. Oh, here. Yeah. Okay. So here we can see a lot of reads, and we can see in the region that it was not a described gene. So we found so far 35 new genes. So another way to analyze our results is the comparison, the in vitro that comes from the bacterial culture in lab, and in vivo from the intestinal loops. So we can see here a group of the genes that are expressed in vivo, but barely expressed in vitro. And also we can see the way that they are expressed, in a cluster that we call operons. So these results are really in the beginning. And what we can find is, with the transcriptomic maps, we can find new genes, we can find also no-coding RNAs, we can find this structure of how these genes are expressed in the operon structure. Also, the differential expression of the C perfringens in vitro versus in vivo is going to at least give us clue and show more how the Clostridium causes necrotic enteritis in chickens. And that's it. And I would like to thank you for your attention and the CPRC and also the Agri-Food Canada cluster. Thank you.